Sorry guys for the technical difficulties, but here we are. We made it. I hope it goes well from here. Well, welcome everybody to this package demo session. I'll be chairing it. My name is Ahmed Lajami. It's my pleasure to introduce Dr. Hedia Tnani. She's a staff scientist at Lieber Institute for Brain Development at Johns Hopkins in Baltimore. She will be talking to us about differential expression analysis. So please, the stage is yours. And feel free to ask questions at any time. And yeah, I'll make sure that they're being answered, I hope. So please.
Thank you, Ahmed, for the nice introduction. Can you hear me well? So I will be presenting the differential expression analysis using Limma and qsvaR. So this will be a package demo about qsvaR. First, let me introduce the importance and the challenges that we face when studying the human brain. When studying and trying to understanding a neuropsychiatric disorder, our studies are conducted on post-mortem brain tissue with an average post-mortem interval that could exceed 12 hours. That's why this could lead to degradation effects in our post-mortem studies, and this can lead to confounding effect in our differential expression analysis. So previous work in our team has led to the conclusion that qsva and quality surrogate variable analysis creates new variable that you can adjust for to capture the degradation associated effects, and this has led to increase the reproducibility and reduce the association with degradation in differential expression analysis in public datasets. This has led to the development of qsvaR to remove the effect of RNA degradation from post-mortem human brain tissue. So qsvaR is already in, you can install it via BioC Manager. And here, so when we will do the demo, you will find the vignette to this package. So the workflow that has led to the development of this package is the following. We have 5 donors, and from these 5 donors, we extracted 6 brain regions, and then we let, it's like tissue degradation on the bench at 4 time points. Then we made like RNA extraction, the sequencing, and we used the SpeakEasy pipeline to get from FASTQ files arranged, summarized experiment object. So here, Nick in the audience, he's the developer of the SpeakEasy pipeline. Please, if you have any question and you want to know more about the SpeakEasy, don't hesitate to ask him questions. And from the arranged, summarized experiments, from these 6 brain regions, so we have merged all these summarized experiment in one degradation matrix. Here in the rows we have the transcripts, and on the columns we have the different samples. Yeah, sorry.
So for the tissue degradation time, that's additional time other than the postmortem interval, or?
Yeah. So after getting the brain tissue, so we let this tissue to degrade. Yeah.
Keep going.
Okay, sorry. We let this tissue to degrade on the bench, additional to the post-mortem interval.
So the idea is to get different degradation times within a single donor.
Yeah, it's to see, it's like how this degradation, actually to model the degradation time, and then to see the interaction between the degradation time and the region. So we applied these 2 models, the main model, which is degradation time, and the interaction model, which adds interaction term of degradation time and region. And then we selected the top 1,000 transcript. We merged this like these two, it's like a top 1000 transcript from the main model and from the interaction model. And if you go, to and you load the library qsvaR, there is a function select transcript where you can choose between different QSV models. The one I presented is the standard model, which is the top 1000 transcript associated with degradation from the main and the interaction model. Then we extended this approach to the top 1500 transcript associated with degradation from the main and the interaction model. And we added to it, there's also another model, which is the cell component model, which involves the convolution of the degradation matrix to determine the proportion of the different cell types. So as I said, the cell component model, so we use the convolution. It consists of deconvolute the bulk RNA-seq using single-cell RNA-seq to get the different cell proportion of the degradation matrix. Here we saw that the cell composition vary by region in the degradation matrix. We can see here the different region that we used and the different cell proportion and the different cell types. So then we added, we adjusted for these cell types in our cell component model. And here in this demo, we will be using brain-seq phase 2 dataset, which is ribo0 RNA-seq data for 900 samples across two brain regions, the dorsolateral prefrontal cortex and the hippocampus brain region. So please, maybe we can now start, loading the dataset and going to the Galaxy platform. So I have to go to the platform. Let me see the history because it was
called workshop.byconducted.org. I think.
It's already on the platform, so maybe I will be using also my R Markdown, but please don't hesitate, to use the platform and let me know if it works. So there's the qsvaR package demo there. So this is the same, the markdown that you will find in the platform. And we will start by loading the different packages that we will need. I did that last night because of all the problems, everyone got during the workshop. So I had this backup plan, So let's load the different packages and then let's load the data. Here, if you want to have and to understand more about the data, here there's a link to it. So please don't hesitate to go to this link, to understand more these data sets. Now, what we will do first is loading the RC object and have a look at and have a general overview of the object. So if you load, this data set, you will see that it's a ranged summarized experiment. Here, it's as Stefano explained, we used the TIG TIG summarized experiment. So that's why you will see this summarized experimentable abstraction. It's about the abstraction layer they were talking about before. So we have here the transcript, the features, the different samples, the TPM because the assay, so this data has been TPM normalized. And then we have all the metadata that you will find here, in the part of the sample information. in the R markdown, there's like a background, about like summarized experiment and ranged summarized experiment. So as you know, ranged summarized experiment is a subclass of the summarized experiment class, which mean that in the rows here, you can use the G ranges and genomic ranges features. So when we will move, so here in the part of data normalization is describing, the how to get the TPM assay. And then, explaining, well, it's to have here how to extract, the TPM assay using tidyverse. We will move to the part of data distribution. So it's to have a look at the distribution of the TPM assay, transcript per million, and then to see, to plot this TPM, so and to see the box plot of TPM for each diagnosis and by region. Here we have control and schizophrenia in this case. So far so good? Could you reach this level? Okay. I will move this like that because we don't need it for the moment. Okay. So we will move to the correlation analysis. It's important to have an idea about the highly correlated variable. Why? Because it can significantly impact the robustness of the model by creating unstable estimate of the variance fractions. So for that, we unlisted, many of the variables because they were in a list format. And then we used the concourse pairs from the variance partition package, which performs a canonical correlation analysis between the pairs of variables. So the concourse pairs function computes the canonical so the concourse pairs function computes the canonical correlation between each pair of variables specified in the formula argument you see here. So in this part, we first define a formula, and then we compute the pairwise correlation between all the variables specified in the formula. You can see it here. So then we will move to create the heat map. For the heat map, we will use complex heat maps. So this is only to have like an idea about the correlation. And once we run, the complex heat map explaining here, so this function and this line explain, the script that has led to this heat map. We see that in the correlation heat map that there is a high correlation between region and total assigned genes, and then the mito-mapped and region, and the mito-mapped and total assigned genes. So one of the strategy for addressing for multicollinearity, when we have highly correlated variables, this leads to a multicollinearity, and one of the strategy to avoid that is removing one of the highly correlated independent variables. That's why, so we had to keep, or we have to keep or mito-mapped or total assigned genes in our model. So before going and moving to the quality surrogate variable analysis, could you understand this part? Is there any question? Actually, this is the most important part now of the analysis, which is the qsvaR and quality surrogate variable analysis. to have an overview of, of this analysis. So first, we will be identifying surrogate variables. Then we will estimate the effects of these surrogate variables. After that, we will adjust for the effect of these variables and perform a differential analysis on adjusted data. For that, in the qsvaR, so we can run this analysis step by step, or we have a wrapper function for that. So as I explained, there are three different qsvaR model: the standard model, the top 1500 model, and the cell component model. We will use in this demo the cell component model because it's the most effective of our models. And we will see in this part how to run the qsva step by step. So if we want to run the qsva step by step, here for a matter of time, we subset the ranged summarized experiment object. So we choose the GLPFC region, and then we, to get the degraded transcript for qsva from the cell component model, we use the getDec_takes function. So for that, we have to specify our ranged summarized experiment and the type of qsva model we want to run. In this case, it's the cell component model. And we will get, after that, we can have a look at the dimension of the data. So since I have it already here, we can have a look at it.
So this is just selecting a predetermined list of transcripts that are known to be degradation associated?
So from the select transcript function, we have a list of like transcripts that are associated with degradation. And this will subset, from your data, from your range summarized experiment object, this selected transcript with their TPM.
So it's just a pre-specified list of transcripts?
Yeah. So to have a look at this, so let's have a look at this object. And you will see here that we have the transcript, the sample, the TPM. What we did, so if we have a look at select transcript and we put cell component, if you try to put here the cell component, cell component. So you will see that we have here the list of transcripts that we've got from our analysis that are associated with degradation. And then, so this getDec transcript, the function, what it does, it's subsetting, this transcript in your ranged summarized experiment. Then we can, so the package we have getPCs, which applies like a principal component analysis of the degraded transcript. And then the most important part, of this analysis is the qsv function. So the qsv function is employed to determine the required number of principal components that are needed to capture the underlying variation, which is, in this case, the degradation in our data. For that, we will need the dec transcript object, the mod. So here we have to create a model matrix for the regression analysis using the predictors, so diagnosis, age, sex, and race from the column, so from the call data of the ranged summarized experiment. And from here, so after the having the model, and here we specify the assay name, the TPM, we can get, the number of QSVs we have to adjust for. So yeah, this function, if you will have to run it on the platform, it will take some time. So be patient. If it's like it takes some minutes for that. And then there is we can run the qsvaR using the qsvaR wrapper function. So it makes easy instead of going step by step. So we have this integrated function, qsvaR. And here, so for that, we have to specify first, well, we have to set seed for reproducibility. Then we have to specify the range summarized experiment object, the type, the qsvaR model we talked about, and then the model and the assay name, in this case, the TPM. Let's have a look at this object. So here you can see that from the QSVs, we've got that we have to adjust for 25 QSVs. And here, so we see that we have in this like qsvaR PCCC, in the column, the different QSVs and values we have to add to our model. We will see that now. So yeah, so here, so it gives us the column from the QSV1 to the QSV25. And once we get this object and these QSVs, so we do a differential expression analysis. Sorry, yeah, go ahead.
What is it using random number generation for here?
This is for reproducibility.
No, I mean, but what is the random component of the algorithm?
Actually, it uses the QQSVs uses the num-SV from the SVA package, and that num-SV has a randomness, so that's why we have to use a set seed. Otherwise, we will get different QSVs we have to adjust for. That's why we have to set seed for that. And then, we do a differential expression analysis. Once you get these QSVs, you don't have, you can run a normal, and usual differential expression analysis. So in the case of the QSVA, we add these QSVs to our statistical model. For that, we will use the cbind function. And as we said, the QSVs adjust for the degradation effects. To run the differential expression analysis, we use Limma, so we extract the transcript expression value, and then we run the standard linear model, so we use lmfit and the ebase function. So the empirical based statistic for differential expression, the ebase, so it improves the stability of the estimate, particularly where the number of samples is small. And after that, here we want to have a look at the column names of the fitted model, and we extract the differential expression results. So we use the top table, then we specify the coefficient. In this case, we are running like case control, so schizophrenia control. That's why we choose this coefficient, DX schizo. We don't filter for p-value, so p-value equal to 1, and then we keep all the genes. We want all the genes to be returned. And we can explore the top results as shown now here. And we can see that we've got our transcript, the log4 change, the t-statistic, the p-value, the adjusted p-value. So here are our results of the differential expression analysis. And then, so it's to show the difference between, when applying qsv and without applying qsv. We have a function, the g= plot or differential expression quality plots. So this function, in this case, so it takes into, here we have the differential expression without adding the QSVs to our model. That's why it's the model without correction. And we give that to the function, and we can have this kind of visualization. So in this visualization without QSVA, we have the degradation t-statistic from the package. And we have here in the x-axis the differential expression t-statistic that we've got, from our a differential expression analysis. We can see the bins here, and the color shows the number of data points in each bin. So if you see blue to purple color, it means that there are few data points in this bin and few transcript. If it goes to green or yellow, it means there are moderate data points. And if it goes to bright yellow, it means that there are a high number of data points and high number of transcripts. So from this plot, we can see the most important, that the correlation is 0.48. So the correlation between the differential expression t-statistic and the degradation t-statistic. And this high correlation could be masking the true differential gene expression between our case control groups. So it's in some way masking the true, biological differences in our differential expression analysis. When we do the same d= plot for the model with QSVs, so we take the significant transcript from our differential expression analysis, we can see that the correlation is almost not, it's close to 0. So it suggests that there is a minimal relationship between degradation and the differential expression results. Do you have a question? Any questions so far? So this plot indicates that the degradation effects have been effectively eliminated from our data. And we can move to the conclusion. I don't know if okay. So for the conclusion, maybe, can I want to show the slides. Sorry for the interruption.
Just go back. Can you exit now?
Okay.
Go to Google Chrome. Here.
Here. Okay. Okay. I will. So as a conclusion, during this workshop, we gained insights and practical knowledge of how to adjust for degradation in post-mortem human brain tissue using qsvaR. The results from the differential expression analysis with and without QSV normalization shown in the 2D plots reveals the effectiveness of degradation effect removal, and the use of QSVA effectively removes degradation effects. And I would like to acknowledge all the team that has been a part of this project and that helped me a lot to understand the qsvaR project. And especially, it's like Louise, Nick, and Josh, who has developed this function of the qsvaR package, and especially Leo for giving me the opportunity to work in this amazing project. And thank you very much.
So thanks, Hedia. So we have one online question. So thanks a lot for the education. What about the trend equal true argument in e-bias? Is it optional in this pipeline in eBay's?
Let me have a look.
This is bulk RNA-seq, right?
Yeah, it's bulk RNA-seq, yeah.
Yeah, I would probably use trend equals true.
Yeah, so
Is it optional?
actually, it's for demo. So, it's like I use the data and I didn't spend too much about it because the main, it's like demo is about, sorry, I don't see myself actually. So the main objective is to show how qsvaR, remove the effect of degradation. But if you have maybe to run like bulk RNA-seq, you have to add this option, to your code.
Yeah, I would say the short answer is just run eBay the way you would normally run it for RNA-seq.
Yeah.
How many minimal samples are required?
Sorry.
The model?
Sorry?
The sample size. The sample size. Minimal sample size.
To run the pipeline. Actually, here we use the 900 sample. But I think you can try run it because we are testing in this moment. I'm testing the package with public data sets. So you can try to run it and see, it's like if it's affected by a minimal sample size. For now, it's like in this demo, we have, if I'm not mistaken, in the data, we have 900 sample in this like demo. But you can try to use it for like, how to say, a smaller sample size and see, if it runs well. But normally,
Well, how do you choose those degradation genes
Mm-hmm.
to get those like SVAs? You choose some genes to calculate those components. You have like 25 components. How do you choose those genes? You have some ground truth or something.
So,
how do you run the SVA? You have some your subsets.
We have from the cell component model, we joined the main and the interaction. So we have 1700 transcript when you run like select transcript.
So how do you get those? So what's the meaning of those cell components?
The QSVs. The QSVs are the principal components that are associated with degradation. So it's like you have, the transcript that are associated with degradation, and it's running like principal component, but for these transcripts that are associated with degradation. That's why you get the QSV that are like principal component analysis PCs, but for degradation effects.
But you have to know those genes. How do you know those?
From the main and interaction model, we ran Limma for that, and we had the main, the degradation time, and the interaction model having the degradation time, with region, the interaction term adding,
One question in the back, please.
I was curious, why did you decide? I mean, I don't know if you said what you guys do, but why did you decide to use TPMs and not raw counts?
Why did we use TPM instead of row counts? because this data, since it's transcript, so it's like TPM normalized, and that's why it came like with TPM. Row counts, it has to be genes. So and genes actually it's good to say it. So when we ran Limma, we didn't use Limma VUM because these are TPM but not like row counts. That's why we didn't use VUM, we used Limma because these are like TPM normalized like data set.
But I don't know what you're processing the data with, but if you're processing with someone, you can also get raw counts, and the authors of Limma don't recommend using TPMs, and they recommend using raw counts.
Yeah, well, in this project, we were more interested in getting the transcripts that are associated with degradation from the pipeline. We've got those transcripts. That's why we use TPM.
So it's RNA-seq.
Yeah, it's RNA-seq,
But
RNA-seq.
If you're already adjusting for surrogate variable in your analysis, does adding a QSV improve your analysis?
Yeah, actually, as you see here, and from the publication, it shows that how adding QSVs, you get less like false positives. So when you add QSVs, there are like some false positive because these are associated with degradation. You remove that and you improve your differential expression analysis.
Yeah, but I mean, like, there's a package like just the regular FDA that catches all the technical
unwanted variation. Yeah, this is actually this is like more specific to degradation. The QSVA is more generic for like batch effect and all unwanted variation. So here, since it's like you have a transcript that are associated with degradation and you apply these SVs or NAM-SV, you get the QSV that are really associated with degradation.
Any other questions? All right. Doesn't seem the case. Well, thank you so much for the great package demo. And I think we have lunch now. So
thank you.